Hello students, I am Dr. Animesh Kumar Sharma, and today's lecture we discuss about concept of vector algebra. In today's lecture we have to cover the certain important topics: elementary transformation, rank of matrix, echelon form of matrix, eigenvalue and eigenvector, properties of eigenvalue and eigenvectors, and concept of vector algebra. All the things are related to vector space and vector algebra. So, our first topic, elementary transformation in matrices. Basically, elementary transformation in any one of the following operation, when we apply on a matrix, then it is said to be elementary transformation or elementary operations. The first important operation is the interchange or exchange of any two row or columns. The second one is the multiplication of the matrix elements of any row or any column by any non-zero number. Third and last elementary transformation part is the addition or subtraction of a scalar quantity, suppose k, times the element of a row or column to the corresponding elements of another row or column. Remember, here scalar quantity k not equal to 0. Symbols to be used for elementary transformation is R with subscript ij. Here Rij represent the interchange or exchange of ith and jth row or ith and jth column. The second one is scalar quantity k with Ri. This represent multiplication of ith or jth row by a non-zero number scalar quantity k. And the third one is Ri plus k Rj, that means addition or sum of k times the jth row to the ith row. So, likewise the corresponding column transformation are denoted by Cij, Kci and Ci plus Kcj respectively. Elementary matrices. A matrix obtained from a unit matrix by subjecting it to any row or column transformation is called an elementary matrix. Another important thing is rank of a matrix. Remember, all these important points are closely related to representation of quantities related to vector algebra. So, rank of a matrix. The rank of a matrix A is said to be small r if it possesses the following properties. The very first property is there is at least one minor of order r which is non-zero. Here minor represent a submatrix or a part of matrix in a square form. Every minor of order greater than r is 0. Rank of a matrix is generally denoted by letter rho of A. This represent rank of matrix A. Some important results related to the rank of matrix. The rank of a matrix remains unchanged, or we say not affected, by elementary transformation, and the rank of the transpose of a matrix is same as that of the original matrix. We know that what is transpose? Transpose is nothing but interchanging the row to column of any given matrix. So, if we find out the rank of any original matrix, and after that if we find the transpose of matrix and then again we try to get the rank, then always both are equal. The third property is the rank of the product or multiplication of two matrices cannot exceed the rank of either matrix. So, in symbols we can represent it as rank of AB less than or equal to rank of A, or we can say rank of AB is less than or equal to rank B as well. Some points you have to remember. First is the rank of matrix is less than or equal to R. Here R represent a positive integer if all R plus 1 rowed minors of the matrix are 0. Next is the rank of a matrix is greater than or equal to R if there is at least R rowed minor of the matrix which is not equal to 0. The rank of a null matrix is 0. Remember, null matrix having all the elements as 0. Null matrix is also called a void matrix. So, the rank of a null matrix is always 0. The rank of a non-singular square matrix is always equal to its order. Let us consider a matrix. Suppose we have a matrix of order 3 by 3. The first row elements are 1, 2 and 3. The second row elements are 3, 4 and 5, and the last row of the matrix is 4, 5 and 6. Now, if you find the determinant of this matrix, then we will get 0. Therefore, rank of matrix A is less than 3. Why? Because if the order of this matrix is 3 by 3, then at this order we get its determinant is 0, and according to the condition and definition of rank of matrix, we have at least one non-zero minor of the given matrix. So, there is at least one minor of this matrix A of order 2, that is 4, 5 and 5, 6. First row element 4, 5 and second row element 5, 6. If we take these sub matrix or sub minor from the given matrix, then definitely its determinant is not equal to 0. So, the rank of the given matrix is equal to 2. Now, another important point related to matrix, echelon form of a matrix. A matrix A is said to be in echelon form if every 0 row of matrix A occurs below a non-zero row and the number of zeros before the first non-zero element in a row is less than the number of such zero in the next row. Remember, echelon form is important to represent the rank number of a matrix. The rank of matrix in echelon form is always equal to the number of non-zero row of the matrix. For example, we have a matrix of order 3 by 4, 3 row and 4 column. The first row element of the matrix are 0, 1, 3 and minus 1. The second row element are 0, 0, 1, 4 and the last row of the matrix A is 0 row. All elements are 0. So, the number of non-zero row in the matrix A is 2. The first row is non-zero. If any one element is non-zero in the corresponding row of the matrix, then it is counted as non-zero row. So, since we have 2 non-zero row, then the rank of this matrix is equal to 2. Any non-zero vector x is said to be a characteristic vector or eigenvector of a matrix A. If there exists a number lambda such that Ax equal to lambda x, where A is a matrix of order n, that is a square matrix, and x is a column vector, that is capital X will be equal to a column matrix, any number of row, but the column should be 1. Then lambda is said to be a eigenvalue or characteristic root or characteristic value or latent root or proper value of the matrix A, whereas capital X represent the eigenvector. The matrix A minus lambda I, this expression is called the characteristic matrix. Lambda is a scalar quantity, I represent the unit matrix and A represent the square matrix of order n. The determinant of this expression, determinant of A minus lambda I, is called the characteristic polynomial of matrix A, and if the equation takes in the form of determinant A minus lambda I will be equal to 0, then it is called the characteristic equation of matrix A. This matrix equation or this characteristic equation is very useful to determine the value of eigenvalue lambda. The determinant of the characteristic equation of a matrix of order 3 can be solved using the following formula. If you want to find the characteristic equation directly from the formula, then it will be easy to take the eigenvalue or to determine the eigenvalue of any given matrix A of order n. The formula is lambda cube minus trace of the matrix into lambda square plus sum of the cofactors of the diagonal elements into lambda minus determinant of A will be equal to 0. Remember, trace of the matrix is nothing but the sum of all the diagonal elements of any given square matrix. Now, Ax will be equal to lambda x. Lambda x can be represented into the form of lambda into I into x, where I be unit matrix of same order as A. So, A minus lambda I into x will be equal to 0, and A minus lambda I will be equal to 0 since eigenvector x quantity not equal to 0. So, determinant of A minus lambda I will be equal to 0 or null matrix representation. So, this is the characteristic equation to find out the eigenvector A minus lambda I into x, where x represents the eigenvector or characteristic vector. Now, we see some important properties of eigenvalue or characteristic roots. The first important property is the sum of the eigenvalue of a given matrix is equal to the sum of elements of principal diagonal, or we can say that the sum of the eigenvalue of a matrix is the sum of the elements of principal diagonal. The second important property is the product of the eigenvalues of a matrix is equal to the determinant of the matrix. And third and important property is, if I is the eigenvalue of matrix A, then eigenvalue of following matrices are given as properties of characteristic vectors or eigenvectors. If x is an eigenvector of the matrix B corresponding to the eigenvalue lambda, then k into x is also an eigenvector of B corresponding to the same eigenvalue lambda, where scalar quantity k is any non-zero scalar. The second property of eigenvector is, if capital X is an eigenvector of any matrix B, then x cannot correspond to more than one eigenvalue of B. Important points related to the matrix theory: if two or more eigenvalues are equal or same, then the corresponding eigenvectors may or may not be not necessarily linearly independent. Second point, the eigenvectors corresponding to distinct eigenvalues of a real symmetric matrix are orthogonal. Any two eigenvector corresponding to two distinct eigenvalue of a unitary matrix are also orthogonal. We start vector algebra. Vector algebra has become an indispensable tool in computational biology, providing a formal way to describe spatial relationships, directional process and multidimensional data. Here is a point-wise breakdown of the concept of vector algebra as applied to computational biology. So, first we see the basic definition of vectors. A vector is a mathematical entity with both magnitude and direction. In biology, vectors can represent numerous phenomena such as gene, expression levels, molecular gradients and movements directions. What is gene? So, the basic concept of gene is, a gene is a unit of heredity in a living organism. It is a segment of DNA that contains the necessary information to make a specific protein or set of proteins. Each gene occupies a specific position on a chromosome. Genes carry the information necessary for the synthesis of proteins and which make up the structure of cells and direct their activities. Basic operations of vectors: addition and subtraction. The most important and most basic binary operation is addition and subtraction, useful in combining the effects of multiple vectors. Scalar multiplication, scaling the magnitude of a vector by multiplying with a scalar. Dot product or scalar product gives a scalar result often used to determine the angle between two vectors or the magnitude of one component of a vector in the direction of another. So, it is very important tool of vector algebra, that is dot product. Cross product gives a vector result used to find a vector perpendicular to two given vectors. So, dot product and cross product having more importance in the operations as used in vector algebra. Hence, genes are fundamental to the function and control of all organisms, including the growth, development and reproduction of the organism. In simpler terms, a gene can be thought of as a set of instructions for building and operating a living organism. Each organism has many genes. Humans have estimated to have around 20,000 to 25,000 genes in their genome. Magnitude and direction, another important property which is used in vector algebra. The magnitude provides the size or length of the vector and the direction tells which way the vector points are. Applications in genomics. Vectors can represent gene expression profiles where each component of the vector represents expression level of a specific gene. Comparing vectors can help in understanding similarities and differences in gene expressions among different conditions or organisms. In structural biology, vectors can represent coordinates of atoms in protein structures. Vector algebra is used to compute geometric features such as angles between amino acids of distances between specific residues. Functional annotations. Gene or protein function can be encoded into binary vectors where each position in the vector indicates the presence or absence of a particular function. Similarity between these vectors can provide insights into functional similarities. Network biology, one of the important aspects of vector algebra, is used in network biology also. Nodes, that is genes or proteins in biological networks, can have associated feature vectors. Vector algebra can then be used for tasks like module, direction or clustering. Differential expression analysis. Using vectors to represent expression levels under different conditions can help in identifying upregulated or downregulated genes. System biology. Modeling biological process or system can involve vectors to represent states of the system. Changes or dynamics in these systems can be studied using vector operations. High dimensional data. Modern biological data is often high dimensional. For example, multi-omics data. Vector representations help in reducing dimensionality and identifying key features in the data. Machine learning and artificial intelligence. Biological data, once converted into vectors, can be fed into machine learning algorithm. This is fundamental for tasks such as classification, clustering or predictive modeling in biology. So, the concept of vector algebra are deeply integrated into many areas of computational biology. The ability to represent complex biological data as vectors and then manipulate and compare these vectors using algebraic operations is fundamental to a vast array of computational technique and analytical methodology in the field. Let's deliver into some simple numerical example based on the previously mentioned concepts of vector algebra in computational biology. So, first we take one basic vector representation. Assume you are studying gene expression levels of 3 genes, suppose gene A, gene B and gene C in a particular organism. So, expression levels might be represented as a vector. The first vector having the element as 5, 7, 2, whereas in the second vector the elements are represented as 6, 8 and 1, where 5, 7 and 2 are expression levels of gene A, B and C respectively, and similarly we have V2 also. So, if we apply the binary operation of addition and subtraction for both the vectors given two different conditions or samples. First sample is control sample, another one is take as treated sample. Control sample elements are represented by V1 having the elements 5, 7 and 2, and treated sample is represented by V2 having the elements 6, 8 and 1. If we take the difference in expression levels, then can be found by subtracting V2 minus V1. So, we have to subtract all the elements one by one from V2 to V1. 6 minus 5 we get 1, 8 minus 7 again we get 1, and the last corresponding element of V2 and V1 are 1 and then minus 2. So, we get minus 1. So, the difference of treated sample and control sample is denoted by V2 minus V1 and having the element 1, 1 and minus 1. The second operation is scalar multiplication. To understand how gene expression levels change when scaled by a factor. For example, if we multiply the control sample element by a scalar quantity 2, then we get 2 into 5 that is 10, 2 into 7 that is 14 and 2 into 2 as 4. So, we have the scale after scalar multiplication of scalar quantity 2, we modified the vector quantity control sample as 10, 14 and 4. The next important operation is dot product. Let us take two vectors, one is V3 and another one is V4. In sample V3 we have 3 elements as 3, 4 and 0, whereas in V4 we have 3 elements 1, minus 1 and 2. So, the dot product or scalar product of these two vectors is represented by V3 dot V4, and V3 dot V4 again we have to multiply the corresponding position element, that is first element of V3 should multiplied with first element of V4, second element of V3 should multiplied by the second corresponding element of V4 and so on. So, we have 3 into 1 first element multiplication of V3 and V4. Similarly, the second element multiplication are 4 into minus 1, and similarly the last element of V3 and last element of V4 having the multiplication as 0 into 2. So, we get 3 minus 4 and plus 0. The resultant is minus 1. This minus 1 represents the dot product of two vector quantity V3 and V4. The dot product can be used to determine the cosine of the angle between two vectors. So, if we want to determine the angle between these two vectors, then this can be used in the form of dot product. Another important part or another important property of binary operation which is used in vector algebra is magnitude. So, if we want to find the magnitude of vector V1, then we have to find out the modulus of the vector V1. How can we determine modulus? Magnitude will be equal to square root or root over all the element square and then sum of all the element. So, we have 3 elements under sample V1, 5, 7 and 2. So, if we want to find out the magnitude, then we have to take root over 5 square plus 7 square plus 2 square. 5 square means 25, 7 square 49, and similarly the last element 2 square will be equal to 4. The sum of all the element is 78. So, root 78 represent the magnitude of the vector sample V1. Now, we have to determine distance between vectors often used in clustering or phylogenetics. So, for vector V1 and V2, the Euclidean distance is defined under distance will be equal to, first we have to take the distance between the corresponding element, after that we have to take the square of difference of element, and then at last we have to add all the resultant values. So, for vectors V1 and V2, we find the distance. So, root over the first element difference are 6 minus 5, and after that we have to take the square. So, we have 6 minus 5 square. Similarly, for second corresponding element of both vector, we have 8 minus 7 square, and the last element of the V1 and V2 are 1 and 2. So, the corresponding difference is 1 minus 2, and again we have to take the square. So, after taking the difference and squaring the resultant of the difference, we get square root of 1 plus 1 plus 1. That means the distance will be equal to root 3. This distance represents the distance between vector v1 and vector v2, and it is also said to be Euclidean distance. Now, functional annotation with binary vectors. Let us say we have two genes and their associated functional annotation A, I indicates the presence of a function and 0 indicates the absence. So, gene 1 represented by suppose F1 and gene 2 represented by suppose F2. So, F1, in F1 we have the samples as 1 0 1 0 has functions 1 and 3, and F2 suppose represented by the elements 1 1 0 0 has functions 1 and 2. Similarly, might be gauged by counting shared functions or calculating consist similarly. Similarity might be gauged by counting shared function or calculating cosine similarity. These are some simplified examples which demonstrate how we apply vector algebra in computational biology also. Gene is the most common thing which is used over the biology. So, these simplified examples, but they illustrate the foundational elements of vector algebra in computational biology. So, vector algebra is also used for matrix concept. Similarly, if we take only one column or if we take only one row in any matrices, then it is also said to be column vector as well as row vector. So, matrix theory along with the vector algebra is very important for computational biology. In computational biology, we have many samples are there, and to construct the system of equation, to analyze the operations, to analyze the binary operation between the sample of vectors, between the sample of matrices, we have to use vector algebra as well. So, as the complexity of the data or the question being asked increases, so too will be the complexity of the operations and algorithms are applied as well.